Hello everyone, I'm Euan, and today I'm going to present some new results from my PhD, which is looking at how well fitness proxies predict the long-term genetic contributions using historical human data. Today, I would like to start with an equation. This is the secondary theorem of natural selection, which explains how we can predict the evolutionary change in a phenotype using the genetic covariance between that phenotype and fitness. This means that having an accurate measure of fitness is vital for understanding how behavioural traits evolve and how animals are responding to selection pressures such as environmental change. Fitness is ultimately a measure of an individual's ability to contribute genes to the population at some future point. However, this is generally impossible to measure. As a result, it is common to use proxies of fitness, something relatively easy to measure but nonetheless closely relates to an individual's fitness. These can include measures of survival, for example lifespan, measures incorporating reproductive success of individuals, for example lifetime reproductive success or LRS, or even measures incorporating the quality of the offspring produced, for example LRS survival to adulthood, which counts only the number of offspring that then went on to survive to reproductive age. Or, finally, number of grandoffspring, which incorporates both the survival and reproductive success of those offspring. Human genealogy records provide a unique opportunity to estimate the genetic contributions of individuals several generations down the line of descent. We can then examine to what extent these four fitness proxies explain the variation in the long-term genetic contributions to evaluate how well these proxies approximate fitness. We predict that these proxies will increasingly predict the long-term genetic contributions as they increasingly encapsulate more information about reproductive success and offspring quality. We used a pedigreed population of humans spanning 4 centuries from 2 parishes from the canton of Glarus, Switzerland, to estimate the genetic contributions historical individuals made to the current population. We estimated the genetic contributions using the method described in Hunter et al. 2019 heredity. Essentially, the method involves estimating the total relatedness each of our historical individuals has to the current population, using the pedigree information. We estimated these genetic contributions to the current population for all individuals born early enough that would allow the estimation of genetic contributions at least 8 generations down the line of descent. This gave us a total of 2,740 individuals born between 1575 and 1734. We then examined how well each fitness proxy predicted the long-term genetic contributions through comparing the R-squared values of 4 generalized mixed models with each fitness proxy fitted in turn, while controlling for potentially confounding factors such as sex, birth parish, the migration of offspring, and birth cohort effects. Here are 4 figures, each showing the proportional genetic contributions of historical individuals to the present-day population in relation to each of their 4 fitness proxies. We found that, in line with predictions, the fitness proxies increasingly predicted the long-term genetic contributions with R-squared values ranging from 13% for lifespan to 56% for number of grandoffspring. Overall, our results indicate that the implications of using different fitness proxies should be carefully considered in behavioural and evolutionary biology, with their limitations appreciated. Of course, we do not advocate that only number of grandoffspring should be used as a proxy for fitness. Instead, we hope that this illustrates the potential limitations of using fitness proxies such as lifespan and LRS, and hope that researchers continue to think carefully about which fitness proxy is appropriate for their study system. Thank you, and here are my acknowledgements.